Thank you.
Okay, so we'll wait, a couple of minutes. But as I said, I'm so glad because you are doing fantastic genetic circuit building, and that's kind of what I really enjoyed. And that's the reason I joined Chris Voigt lab, because he at that time, basically doing the genetic circuit building and refactoring at the time. And that's the only reason I joined him, among other options I had at the time. So Lingchong, I mean, you are your office or home, where you are?
I'm in my office.
Oh, okay.
Yeah, I think I'll try to get into office every day. Some sort of normalcy. I think that's useful. At home, I say I work from home, but it's always quite distracting. So working in the office is actually more relaxing.
Right, exactly. So your school is right now fully in-person teaching?
It's in-person teaching. We have to wear masks. I think that's still better than actually giving lectures over Zoom. So it's a little bit of challenge, when you wear a mask for more than 1 hour, it gets a little bit itchy.
Yeah, same here. I mean, we still teaching. We actually teaching the in-person, and then I need to wear mask all the time. So that's kind of thing. But still, that's better than online teaching, I guess.
Yeah. Same here. We also fully in person, but I feel great to see the student in person for the teaching, and it's so different than the online teaching. So
the worst thing of the online thing, even this seminar series, we cannot see the reaction of audience. So that's the worst thing. But another good thing is we basically doing recording. In that way, we could reach to even broader community, and that's why even Africa people and Asian people, right now midnight and so on, but they could watch this one. And believe it or not, I got a lot of email from Africa asking how to watch this one, and then I basically shared the link with them. So I'm so glad, because now this one really become the global event.
Right.
And so
basically, can you tell us about the genetic circuit modeling day? I didn't know that. I just learned that from your email. Why is that something new, or the genetic circuit modeling day?
Oh, so the I kind of named the in every single day that related to the speaker. So for example, today, both of you expert in genetic circuit modeling. So I call that genetic circuit modeling, and we'll have some other genetic circuit people later on. So I kind of specifically include modeling in the name of the day. So the last last couple of weeks, before, I also call it medicine or health day, because they kind of expert in the health and so on. And I kind of every time I kind of try to come up with some name. So so if you don't agree, I can change. I can change.
I think it's wonderful. It's quite challenging to actually come up with one of these terms actually to that's a fit to specific topics.
Right. But every time I kind of try to think, miraculously kind of something come up, and then there are some common theme, and that's why I so delighted to have both of you and then many of the speakers previously. Okay, I guess we need to start. So okay, so good morning, afternoon, evening. Hola, hello, bonjour, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you for joining us, the 10th seminar and genetic circuit modeling day. Very special day to me and my parents in Korea, right now, their wedding anniversary. So congratulations to them. And I thank you for giving me the life, although you may be sleeping right now. I'm also celebrating our 10th seminar by taking COVID booster shot 2 days ago. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Dr. Lingchong Yu. He is a professor at Duke University and trained by Professor Francis Arnold at Caltech. So he is my academic uncle. In fact, his 2004 Nature paper with Francis Arnold titled Programmed Population Controlled by Cell-Cell Communication and Regulated Killing is one of my favorite papers that I read during my PhD training, making me more interested in synthetic biology. So thank you, Dr. Yu. The virtual pro is all yours.
Well, thank you so much. First, I would like to commend Professor Moon for his tireless effort in putting this series together. This is a tremendous service for the community of young investigators, and especially during this challenging time. I really don't know to what extent I deserve the term pioneer, but I will just reflect some of my experiences working in synthetic biology. 20 years ago, that's when I became interested in synthetic biology, when that's roughly the start of the synthetic biology. And time really flies. When I started, my son was just born, and now he's started in college. I always feel like there's something tremendously energetic about this field. Ever since the earlier days, I always wonder what's so special about this field. From my perspective, I came from the background of chemical engineering and did mostly computation for my PhD. Looking back, I was thinking about what's drawn me into synthetic biology. I think it's this notion that you can actually somehow program the cells in such a way that it behave interesting or potentially useful manner. That thing is intrinsically appealing to me. And also in the earlier days, all of the synthetic biology is done kind of like in bacteria. Coming from a background of computation, I told myself, well, okay, that's something, maybe I can do it. So that's how I got started in synthetic biology. The initial motivation was very simple, is a sense of that ability to do something, to control the cells. That it's intrinsically interesting. Actually, earlier days, the synthetic biology is largely focused on capability, in the sense that, can we build some sort of logic gates? Can we exhaust what a combination of logic gates in single cells or in multiple cells? Can we program oscillations? Can we program bistable switches? Or can we generate some sort of collective behavior in a population of cells? It's mostly about to what extent I can do it. I think that's incredibly important, because having the capability is the foundation for us to expand in our imagination. Once we can do something, we can say how we can make this more useful. In fact, actually from this series, and you're gonna hear a lot of wonderful applications of synthetic biology. And many of them actually can trace back to some sort of fundamental devices developed in the earlier days. If I look back, the applications could include, we heard a lot about energy, environment, understanding biology, or computation, and medicine as well, right? Now, if I think about the field, let's think about the reality check, how much our capability has advanced. I have to admit, I feel like sometimes our imagination probably has outpaced our capability. At some point, the kind of applications we can do will still be fundamentally limited by the kind of capability we can demonstrate. How do we judge this capability? We can pat ourselves, we say we're doing a good job. It's hard to come up a benchmark to say, oh, we're doing something great, right? But one way to evaluate this could be the following. This could be a challenge that posed to the community, especially the young investigators. Let's say if we have a team of IGEM team, undergrad students or high school students, you form the team. If you can build something from scratch, from design to construction to demonstration, during that period, during one summer, I feel like we can do it as a community. If I apply that standard, there are certain things we can do that already. For example, if we're building a logic gate, starting from design to the finish line, demonstrating that you can do it, AND gate, OR gate, or whatnot. I feel like I'm confident that with a team of capable undergrad students, they can start to, during one cycle of IGM competition, they can actually achieve that. Some other things, I think it's still out of our reach. There is proof of principle of building oscillators. There is proof of principle of building self-organized patterns. But I think for most of the teams that during one summer, it's probably very, very difficult. It's unlikely for a team of typical undergrad students to be able to go from conceptualization to the point of demonstration of a functional device but during the summer. I feel that is a potential benchmark for the field, that we have really mastered the capability of predictable design of some function, right? So what could be the challenges? There are many. Just without taking too much time, I just have one slide. That's reflecting my thinking. What are the fundamental challenges that we have to overcome in order to advance in this capability. So this is my single slide, thinking about advancing the capability. Fundamental objective of synthetic biology, doesn't matter what applications you have in mind, is about predictive design and implementation. The ultimate goal is that we want to have precision in the consequence of the control. That if we build a circuit, we want the function to be what we want. Now, there is tremendous advance in terms of the precision in the action of control. For example, I can build a circuit, can verify the circuit, can do that in a very large scale with proper techniques like DNA synthesis or like the modern cloning techniques or gene editing, whatnot, right? That's the precision in action of control. And this is actually a huge gap. And two of the fundamental challenges I feel like that require tremendous participation from young investigators. One, I feel like is timing and harnessing complexity. So sometimes it's bad, sometimes you can utilize this complexity. By complexity, I mean the increase in the dimensionality in a system that you're trying to manage. When we think about a circuit, we say 10 genes. It's not only 10 components. It's operating in the context of, let's say in E. coli, 4,000 other genes. So there's tremendous complexity in the dimensionality. Another example of this complexity is in the context of microbiome. if I think about the microbiome, the human gut microbiome, if you have hundreds of up to 1,000 different species interacting with each other, so how do we control this kind of microbiome dynamics, right? The second one, this has been an active research area for many years, roughly the same duration for the synthetic biology in terms of on the experimental side, is timing and harnessing variability. For example, in the context of non-genetic variability, gene expression noise, or genetic variability where you have mutations. Direct evolution is actually a perfect example where we are trying to take advantage of the variability to give us something that we want. Judging from the lineup of the speakers, I'm sure you're gonna hear a lot about the different strategies that try to tackle these challenges from different angles. So with that, I'll stop here. Well, thank you so much. I'm looking forward to Dr. Tian's presentation.
Wow, brilliant talk and inspiring insight. I completely agree with you, Dr. Yu. And I also share the same motivation, excitement regarding genetic circuit and their applications. This is a very relevant because you also mentioned iGEM, and because the iGEM Jamboree event is coming up in November. So my seminar actually series will have a few weeks of break to celebrate the young people's exciting competition event. So again, thank you so much. I mean, your insightful, remark, and that's the brilliant.
Well, thank you.
Okay, thank you. So, so now the main speaker of today with a little bit longer introduction. Dr. Xiaojun Tian received his PhD degree in systems biology from Nanjing University in 2012 and spent 5 years as a postdoctoral fellow at Virginia Tech and the University of Pittsburgh. In 2017, he joined the School of Biological and Health Systems Engineering at Arizona State University to start his lab and synthetic biology research. His research interests are in employing quantitative experiments and mathematical modeling to understand fundamental problems in synthetic biology. Currently, his lab is focusing on the interaction between synthetic gene circuit and host cell, which could significantly affect the robustness of circuit functions. Over the past 4 years, his lab has been investigating the functional loss mechanisms of the gene circuit due to their interaction with the host cell and developing control strategies to enhance circuit functions. His lab has made outstanding achievement with several publications at Nature Chem Bio, Nature Communication, and ACS Synthetic Biology. In addition, he recently received the NIH Maximizing Investigators Research Award, aka MIRA Award, and this is a tremendous honor to any researcher, young investigator. Personally, I love his recent Nature Communication paper titled "Winner Takes All Resource Competition Redirects, Cascading Cell-Fate Transitions," and of course, ABBA's song, "The Winner Takes It All," although both are unrelated, I guess. And Dr. Tian, take it all, take it away. Thank you so much.
Thank you. Let me share my slide. Thank you very much for Dr. Mo's very nice introduction, and Dr. Yu for the very insightful pioneer talk. I think my talk is very related to the several points Dr. Yu covered. Today I will talk about understanding, predicting, and control of circuit host interactions toward engineering robust in circuit. So, and this is my lab. I started my lab like about 4 years ago, and I joined ASU, and I decided to do some research in the synthetic biology. I'm a kind of a system biologist turned to a synthetic biologist. So, and one of the reasons I do the synthetic biology is that because the system biology and the synthetic biology are kind of very close friends, right? So we shared a lot of methodologies, the principles, right? So we combine the mathematical modeling and the quantitative experiment to try to understand the basic science, try to optimize biomedical intervention. So today I will cover our research in the field of synthetic biology. I want to start my story with my postdoc work about the epithelial to mesenchymal transitions. This is the cell phenotype transition from epithelial cells to the mesenchymal cells. Epithelial cells has a tight cell-cell adhesion junction, so they fall, they are connected to each other, they don't move. And the mesenchymal cells will lose all of these adhesions or junctions, so they will become individual cells and they can move. They are very invasive, right? So, and I built the first mathematical model for this process based on this gene regulated network. And through analysis of this model, I predicted that in addition to this epithelial and mesenchymal state, we also have a state in between that is partially empty state. So I proposed a mechanism that we called cascading basketball switches. So basically we have 2 switches, they are coupled. So the cell will transition from epithelial to the partially empty state with the first basketball switch, and then partially empty convert to mesenchymal through the second switch, right? So one of the motivation for me to do the synthetic gene circuit synthetic biology study is that I want to build a synthetic cascading basketball switches. As you know, because even our mathematical model for the EMT process is verified by our experiment, right? But the model is actually very simplified. We ignored many regulators in the process. Our model can generate cascading basketball switches, but we don't know if other regulators is necessary, right? If we can build a synthetic gene circuit with similar gene network, so then we can show, yeah, the design principle of the cascading basketball switch is like this. So this is our design. So basically we have 2 modules. Each of the module is a basketball switch, so they also regulate each other. Here we consider a self-activation module, 2 self-activation modules. So the first module is self-activation of ROC, ROC. So the ROC will promote the production of itself. The second module is a LuxR, it will promote itself. So these 2 modules connect to each other, they promote each other, right? So what we expect to see is when we increase the inducer dose, like the Arabano's dose, for the first module, we could see a cell transition from no color to red or green color. Here I show red only, right? And then if we continue to increase the dose of the inducer, we will see both modules activated. So we see the yellow color. So this is our expectation, right? So actually, if we want to make this circuit work, we have 3 steps. Step 1, we make the module 1, the ROC module as a basketball switch. The step 2, we make the LuxR module as a basketball switch. And if we want the cell fit transition in a stepwise way, so the third step, we may need to tune the connection between these 2 switches, right? So all the story start with this motivation and this gene circuit design, okay? So, and also I want to talk about the modularity. Modularity is very important for the system biology, for the synthetic biology as well, right? So if we study one module individually, and also we can test this module individually and make sure that it works very well. And if we have multiple tested module and we put them together, they are interact with each other, we hopefully the whole circuit will work as expected, right? And but sometimes, or in many cases, this is not the way we are expecting. So the challenge, as James Collins said, it takes months, if not a year or years, to get the network to behave. You have to do awful lots of tweaking to get your network to work the way you want, right? So even we test the modules, we test the paths, and then we put them together, the circuit often don't work, don't function as expected, right? So why we have such a big challenge? What is the problem, right? So another thing I want to talk about is about the design, build, test cycle. This is engineering principles in the CSI gene circuit. I know this, but I didn't know that how many cycles we needed to finish one project, right? Until I see this figure, right? So this is a simplified version about one project to produce a methanic assay. And from the initial idea to the final product, we have to change the circuit many times until it works, right? So what is the problem? One of the bigger problem is interactions between the gene circuit and the host cell. We could have the host cell will have burden because we put a lot of gene circuit into the host cell. And we could have a growth feedback between the host cell growth and the gene circuit. And the gene circuit could compete for the limited resources like the iron polymerase, like the ribosome. So it is very complicated. And usually we don't consider all of these circuit interactions. We try many ways, try very hard to make our circuit work, change the promoter strength, RBS strength, all of this, right? But this is, I want to say this is a fundamental problem we have to do, and we want to know how this circuit host interaction change our gene circuit and how we can control this. That is, okay, so the first work I want to introduce is our first work published for the synthetic biology field. Is about the topology-dependent interference of the synthetic gene circuit function by growth feedback. Actually, this is the study about the first module in our design for the cascading basketball switches. And we tested the promoter for the p-bad promoter for the first module for the ROC, and we see this is ultra sensitive. That is what we wanted, and we say if we close this loop, make a circuit as a self-activation with modeling, and we predict that this is a good design for a basketball switch, right? So this is a very simple design. We hope this will work, and we use the flow cytometry and test this circuit, and we found that yes, it's very good bimodal distribution, right? We say clearly two cell fit. That's great, right? It's a good sign. But when we try to further test this using to do the hysteresis experiment, right? We have for the black curve, the cell initially is off. That means with low level of GFP. And for the red curve, we first to activate the cell, so we have high GFP, and then dilute the cell into the fresh media with various of concentration of inducer arabinose. And we expect to see there is a region, so we could have on and off cell under the same concentration of the inducer, but we didn't see any difference between these two lines. That means no hysteresis, and no memory for this circuit. It is very strange. And we tried many ways to try to make this circuit. We changed the promoter, RBS. We did many ways, but still we didn't see the hysteresis. So what is the problem, right? And then while we are troubleshooting our circuit, simple circuit, and I remembered that I read one paper about from Lingchong You's work, and that's a very interesting work. And they had one circuit. It's also a self-activation circuit. This circuit has no ultrasensitivity, so they didn't expect to see the bimodal behavior, but they did see that in experiment. So they want to know why, and they found that that is because of the growth feedback. So the circuit expression will inhibit the growth of the host cell. So the growth of the host cell will dilute the expression of gene circuit. So there is a growth feedback here. If we consider the growth feedback, we could the black curve is the production sequence rate of the circuit, and the degradation or the dilution curve will be bent, so we could have 3 intersection points, so the circuit is stable, right? So my experiment is more like opposite of this finding. So we found that the memory is lost, and Dr. You's work shows that this growth feedback could provide our sensitivity to the circuit, so the circuit is stable. So, and to find out the reason, we test the dynamics, right? We want to know why initially the GFP is high, but somehow the memory is lost. We tested the dynamics, and we found that after loading the cell into the fresh media, the GFP level decreased quickly to a very low level. So here, the memory is lost, and then we try to use different concentration of the inducer, and the circuit reach to different levels. So the memory is lost for the most of the dose, right? So that is the reason. So the reason is the growth mediated feedback, right? And we can also see that using the microfluidics. And initially, we can see we have one cell with a high level of GFP, and the cell divided to 2 cells. We see the GFP level it decreases a lot, and then we don't see the GFP with the cell continue to divide, right? And even we change the media, from fracture media to the conditioned media, we don't see the recover back of the GFP. The GFP is recovered back because of a high level of the inducer in this case, right? So, and then we think about that, yeah, we needed to revise our model by considering the growth of the host cell. So, and we consider this growth feedback, and the circuit will inhibit, cause some burden, will inhibit the circuit growth. The growth will be slower, and the growth of cell will also cause the dilution of the gene circuit. And we revised our model. We did see we could lose the memory for the low dose of arabinose, right? And here is the mechanism. I really like this demonstration. So here on the left panel, we have 2 lines. One line is the production rate, and the red line is degradation rate. So we have 3 intersections. We have 2 stable, 1 unstable, right? Initially, the cell is here with a high level of ROC, right? And then if we dilute the cell into the fresh media, and the growth of the cell or division of the cell will dilute the gene expression, and the ROC level will decrease. So I can see, let's see, like this. The first cell division, and the cell try to recover back. After the second cell division, the ROC level already crossed the separatrix So this dashed line. So if the system already crosses this separatrix, there's no way to get it back. It will convert to this off state eventually. That's the reason we didn't see the hysteresis, right? That's because of the growth feedback. And then how to solve this problem? We came up with one simple protocol. And instead of diluting the cell into a fresh media, we dilute them into the conditioned media, which is from the media from the stationary phase, in which we don't see the growth of the cell, and we see the GFP level is maintained very well, and we finally see the very good hysteresis curve. We will see the festival region in here, right? So, and then we go back to check the first work for the toggle switch, right? This is very interesting. Here, the protocol is they diluted the cell into the fresh media every 6 or 8 hours to make sure the cell are growing in the log phase, right? And we really want to test the toggle switch with our protocol. We dilute the cell into a fresh media, and that's it. We don't dilute again. And we want to see if the circuit toggle switch also have problem of losing memory, right? And we did this, and we diluted the cell into the fresh media for the toggle switch, and we did see a quick decrease of the GFP level. It's a quick drop, right? This is similar like the self-addition circuit, but the difference is it doesn't depend on the inducer concentration. They all recover back to the initial on state. We can say they all recover back to the on state. Of course, and also the toggle switch, the GFP level maintained very well if the cell is diluted into the conditioned media here. So there's no big difference if we dilute the cell into the fresh media or the conditioned media. If we don't have the cell growth or don't have the cell growth, we always see the hysteresis for the toggle switch. That means this toggle switch is more robust, right? It's refractory to the growth feedback. So what is the reason, right? We want to know. Here we use mathematical modeling to demonstrate this. Here we show the knucklings of the toggle switch because we have 2 variables, and we have 3 intersection points, 2 stable steady state, 1 unstable steady state, and this is the separatrix, right? And initially the cell is here in the high level of lac I, right? And if we dilute the cell to the fresh media, the lac I level and the tight R level will be diluted at the same time. So this dilution direction will be more parallel to this separatrix. And here I will show you. Dilution, try to recover, dilution, try to recover. After several rounds of cell divisions, the cell are still in this region. The cell hasn't crossed the separatrix because it tried to, I mean, the dilution direction is more parallel instead of, here I can show the summary. So we have 2 systems, both could work as a basketball switch, self-addition, and a toggle switch. The difference is the topology. So same function, different topologies, right? So for the self-addition, this dilution direction is perpendicular to the separatrix. So it will lose the memory very quickly. After 2 cell divisions, it will already cross the separatrix and convert to the off state, right? But for the toggle switch, it is more parallel. So after several rounds of cell division, it is still in this domain. It haven't crossed the separatrix. That's the reason it is very robust. Okay, so this is the first work. And following this work, we during the pandemic, we did another work. The students who did this work. And in this work, what do we change? We changed the nutrition, and this nutrition could modulate the growth feedback, and also could change the circuit dynamics. and we did find that. We found the emergent depth oscillations, right? So we changed the LB concentration or the fraction in the culture media, and with 100% of the LB, we say the cell grew very well, and with 0% of LB, the cell doesn't grow right, and for the 100% LB, the GFP level decrease and reach to the steady state because it's the high level of the inducer. And then, but if we change the LB concentration to 20%, we did see the quick drop of the GFP and then a very large overshoot of GFP. And then system relax to the steady state. So the dynamics is completely different from what we did in the first tool. So what is the mechanism? And to understand this, we revised our model by considering the ribosome. So this ribosome is controlled by the cell growth because the cell growth needs the ribosome. But this ribosome will competed by the gene circuit and the host cell. Okay, so that is the only difference. And we use this model to, we can understand why we have this oscillation. And also we make a prediction with this model. And if we change the nutrition systematically from 0% to 100%, and in the model, we just change the Nmax, right? So the maximum number of the cell. So we did say the valley and the peak sometimes, right? So the valley over the curve always decrease with Nmax. So if we have a very high good nutrition, we have a very deep valley, but we only see some peak in some range of the Nmax. So, and the largest peak is found in the moderate Nmax. So that means moderate nutrition, and we tested that. And we changed the LB concentration from 0% to the 100%. And we did see, we only found this emergent depth oscillation in some range, and the maximum peak is found about 10% of the LB, right? Okay, this is a small work following that work. And the third work I want to talk about is about the one-tick-all resource competition, as mentioned by Professor Moon. And this is about the whole circuit, of the cascading basketball switch. So here we have two modules, right? I already introduced this. I think I can skip this. So, and but I want to make to demonstrate this circuit works. At least it's using mathematical modeling, right? So we can consider this circuit like we have two modules. Both modules are basketball switches, and they regulate each other, right? And we can plot the knuckling for each module. And for the first one, we can consider the module 2 as the input for the module 1. So this is knuckling for the module 1. So when we increase the module, when the module 2 level is increased to some threshold, we have a threshold Sn, the module 1 will be activated. So this is the same for the module 2 as well, right? So we have two knucklings, we have 3 intersection points, and here because the link from the module 2 to the module 1 is stronger. So we have the threshold Sn1 is relatively smaller than Sn2. So we have the self fade from the transition from the no color to the green and to the yellow. The yellow means co-activation, right? So, but if we have the stronger link from the module 1 to the module 2, we have something different. We could have the self-fade transition from no color to the red to a co-activation yellow, right? So this is a good design. at least with the simulation, right? We can see this good design. We can see the successive cell-fit transition, right? But what we did, what we found in the experiment is that we changed the inducer dose from 0 to a very large dose, right? And we see initially cell is with low GFP, low RFP, no color, and then we see the module 2 is activated with high level of RFP, and then we continue to increase the Arrhenius dose, and we see just a little bit cells are in this co-activation, no many cells, right? Most of cells are jumped to the module 1 activated cell state, and if we continue to increase, most of the cell will jump to this module 1. That means we always only have one module activated. That is more like a winners. We always have one winner. It's more like a winner takes all. We didn't see many cells in the co-activation region, right? So this is what we expected. And this is what we found in reality, right? So, and we want to know what is the reason, right? And the reason is resource competition. So now we have two modules. These two modules we competed for the limited iron polymerase, the ribosome, and then they will inhibit each other indirectly by competing for the resources, right? So we revised our model, and then we see that the knuckling is changed. So here, when the module 2 increase, we still have the threshold for the activation of the module 1, but if the module 2 is level is too high, it also can turn off the module 1. So same for the module, the knuckling of module 2. So here we have 3 different intersection points: no color, green only, or red only, right? So that is the reason we got no co-activation in the experiment, right? Also, we think about maybe that is because of the mutual activation of the two circuits, right? If we remove these two links, so now we have 2 separate switches. So there's no direct interaction at all. The only interaction could be the resource competition, right? Here we have 2 inducers, Arrhenius and the C6 for the module 2. And then we fix the Arrhenius dose. This dose is able to activate the module 1 if we don't have C6. But when we increase the C6 dose, we see the module 2 is activated. We could see the co-activation, but if we continue to increase the C6 dose, we see most of the cells are in the module 2 activated state. So that means we could have the winner takes all behavior in this 2 separate circuit as well, right? So here, where we have this co-activation, that means that is because with this level of the C6 and Arrhenius, the 2 modules could activate at the same time, so we could reach to the co-activation. But if these 2 doses are not balanced well, we only have one winner. In the circuit of the cascading festival switch, because one module always depends on the other, we only have one inducer, so we will not able to make it work because of a resource competition. But we tried to make it work, and then we did some literature research and found a very great work from Lingchong's lab, right? So in this paper, they talk about we could use a division of labor strategy to enhance the overall productivities, right? So, I mean, yeah, if we use this strategy to solve our problem, we did that. And we put the 2 modules into 2 different cells, module 1 in the string 1, module 2 in the string 2. And these 2 circuits are still connected. And because the link from module 1 to the module 2 is is connected with cross sensing signals. But here we don't have the original design. We don't have another cross sensing from the module 2 to the module 1, but the circuit still works as a cascading backdoor switch. Here is the data. Here, because two cells are in different cells, so they don't have a resource competition problem, right? And then we increase our bundle dose. What we can see first, the module 2 is activated, and then the rest of cell we have the module 1 activated. If we continue increase the dose, we say this two population is stable, right? That means this strategy it worked, and we have the stepwise activation of the two switches. Here, because we have two modules in the separate cells, so we don't see the yellow color, but we did see the co-activation. This co-activation is seen from the two population. Okay, so and following that work, we another student, Austin Stone from in the lab, proposed a different strategy, try to control this winner-takes-all resource competition. Okay, so we use the two self-activation module as one example. And if we don't have a resource competition, these two modules are independent. So we have a very large region of co-activation. But when we have the resource competition, this co-activation is shrink a lot. I mean, it's a very small region, right? And in the field, we have some controllers, the negative feedback controllers, and one of them is a local controller. The local controller is that each module will generate something to inhibit itself. They are separated feedbacks. So this controller works good, and we can see the co-activation region is recovered back to some level, right? And also we have a global controller. This global controller, both modules generate the same thing that will inhibit all of the modules. And we see here, actually, this global controller is not good to control this winner-takes-all. And what our controller is this one. We call this a negative competitive regulation NCR module. And in this module, in this controller, we use DCAS9 and CRISPR system, right? So each module will generate one small get RNA, and the two get RNA will compete for the limited DCAS9 for the inhibition of the modules. So we cannot use a competition against competition. So this design gives us something like this design will punish the winner module, because suppose we have the module 2 is winning, right? It will generate a high level of small get RNA 2. It will compete for the DCAS9, and we have no DCAS9 for the first module. So this module will have no much inhibition. So we give the low activity module some competitive advantage, right? And that's the reason we have a the largest region comparing with the local controller and the global controller. And this is simulation, okay? And we are going to test that in experiment. And the last work I want to mention is about the gene expression noise. We suppose the resource competition could change the gene expression. And we use a very simple system. We have two independent genes, but they could connect it because they competed for the iron polymerase and the ribosome. And we did a simulation and found that when we consider resource competition, we have negatively correlation between the GFP and RFP, and this is a distribution. And if we don't, the reason is that because if we have a fluctuation in the GFP mRNA, it will compete for the ribosome. So that's the reason we could also have some fluctuation in the RFP because of this, right? So we did a simulation, we can remove this fluctuation from the opposite mRNA and this is a simulation. And in this case, we don't see the negative correlation, and it's a wrong correlation. But the interesting thing is that here, actually, if we have the resource computation, the noise is larger if we compare these two cases. And also you can see from this one-dimensional distribution, and if we have the resource computation, the distribution is wider, right? That means we have a larger noise. That means resource computation could add noise into the system, right? So to understand what is that noise from the resource computation, we analytically solve the system for the noise. The noise here, we give the noise for the GFP level. We have 3 components actually. So the first component is from depending on the mean number of the protein. It represents the noise from the birth and death of the protein, right? And the second component depends on the mean level of the GFP mRNA. So it represents noise from gene own mRNA. So these 2 terms is the same as published work, but we have additional the third term. This term depends only on the mean level of the RP mRNA, so the oppositing mRNA. So this whole term is the source of the resource computation noise. And we compared 2 cases. One case is unlimited resources. We usually use this assume unlimited resources, and we for the modeling, right? And here is a case we consider resource computation. And what we can see is that if we compare this, the total noise of the, and we can see this dashed line is a case with resource computation. We can see this dashed line is always below the purple line, the pink line, right? That means resource computation make the noise smaller, right? And in the previous slide, we say this the noise could we could add the resource computation could add the noise, right? So why the resource computation add noise? Because we have a third term, right? And why the resource computation decrease the noise? Because we can see here this red curve. If we have unlimited resources, this red curve is a second term, it's constant. But if we have resource computation, if we have unlimited resources, this level could be decreased. That is the reason the resource computation has a double-edged rule in the gene expression noise. Okay, and also we compared about 25 feedback controller types using the NCR local and global controller, and we compared that, and we found that NCR controller also did a very good job to control this noise. And this work is on the review. And here is summary. Basically, I talked about the 5 stories. The first story talked about the topology-dependent mechanism, on how the growth feedback will affect our gene cycle. And also we talked about if we have multiple modules in one cell, we could have a winner-takes-all behavior. We demonstrated the emergent oscillation that is modulated by the growth feedback if the nutrition level is, and also we propose a new controller, negative competitive regulation controller. And also lastly, we talked about the noise, double-edged. Okay, with that, I want to thank the people in my lab. Dr. Wong did most of the experiment, Juan did the oscillation work, Han did and Austin did the noise work, and Austin proposed the NCR controller. And I also want to thank the people in Xiaojun lab. They helped us a lot when we built the lab. And Dr. Lai for the noise project, and Dr. Wong for the oscillation project. And also I want to thank NSF and NIH for the funding. Thank you very much. And with that, I'm happy to take any questions.
Oh, fantastic talk. That's brilliant. I love it. So I especially enjoyed this talk because I personally have worked on similar project for, I guess, more than a decade since my postdoc year in 2010 with the Chris Voigt. And I also love to see the mathematics regarding noise. I mean, nowadays, I mean, not many papers are discussing this important aspect of biology, of course, compared to the period between, I think, the 2000 and 2015 or something like that. So I already see one question from John, and he said, very exciting and important work. My question is, many of your conclusions stem from either rapid cell division or uneasily reachable ceiling of resource expression, which are both particularly true in bacteria, but not less so in eukaryote. Do you think that building circuit in mammalian cell, for example, would encounter similar types of problems that you describe, or do you think the challenges there will be in mostly of a different category? That's interesting question.
That's a very great question. Actually, here, because of limited time, I didn't mention that many other great works published by other labs. So actually, we do have the resource competition in the mammalian cells. It is published in the Nature Communication, and the behavior could be different. Maybe we have sufficient ribosome, but we have very limited iron polymerase. I don't remember the details, but we do have the resource competition in the mammalian cell. When we design gene circuit, we needed to think about that as well, right? So yeah, for the growth feedback, we always have the growth for the mammalian cell and the bacteria. And when we have one circuit tested very well in the lab, and we try to apply that in a different condition, the growth could be changed. Maybe our circuit also will be changed, behavior will be changed. So that is a bigger problem. That's the reason we want to know why some circuit could be very robust, but not the other. We give a very simple circuit as a proof of demonstration, but when we have a different circuit, we want to know maybe we have an optimal design or something, right?
Okay, wonderful. So I have another question from Feng King. Excellent work. Congratulations on your, of course, NIH award. Also, congratulations from me. And for your growth-mediated memory loss project, I see the toggle switch can bounce back even after dilution, but not for your positive feedback circuit. Do you think it is possible because of the intrinsic difference between a toggle switch and positive feedback, even though they are generally both bistable circuit? Also, I guess, I mean, I probably gonna answer that question first. Probably he asking or not, or she. Also, okay. You use the RRC as a regulator. I'm wondering whether the memory loss is related to or specific to the RRC, since RRC and arabinose is part of the E. coli gene network. Interesting.
That's a good question. And yeah, we use different genes for the circuit. That's could be something unfair or for the comparison. But for the string, we did use some mutation strings, so I guess that is not a problem. So if we use arabinose or something, and what we want to show is actually, as I showed in the why the self-activation module is more easy to lose the memory, because dilution direction is more go against directly the separatrix, but for the toggle switch is more parallel, so it's not easy to cross the separatrix. So I think that is the most important reason, but could be from the gene specific or something. Yeah. But thanks for your question.
Okay, so I'm checking any more questions. Actually, I wanted to ask some question, but we already hit the 11:04 in my time. Let's see, one more time. I'm checking. Okay, so we don't have any questions. So I'll ask my question during the follow-up session. So, to conclude, thank you all for joining and staying today again. And in fact, today is a very important day for synthetic biology. Actually, many of our colleagues are discussing the future and vision of synthetic biology at the EBRC Council meeting right now. And I need to join it after this seminar series to continue our two-day exciting discussion. And additionally, Ginkgo Biowork, actually, as you know, Ginkgo is one of the leading synthetic biology, companies, and I have known all of the founders, Tom, Jason, Reshima, Barry, and Austin since 2006, I think. Thanks to our weekly SynBio working group meetings at MIT and SynBYSS meetings, and Ginkgo's annual conference called Ferment is actually also happening right now in Boston. So that means, very important, 2 meetings actually going on today and this seminar series, and this is the now 10th seminar. So congrats, Dr. Tian, and also congrats Ginkgo on your huge success and going public. And I wish best of the luck for centuries, and of course, and that this is again, the 10th seminar, and we will have a long 3-week break until November 18 due to iGEM Jamboree competition in November and my 10-day seminar trip at 4 universities and 3 conference, including AICHE meeting. So if you are attending AICHE meeting or living in Boston, New York, or Washington, DC, let's meet there in person. And on November 18, we will have Dr. Humin Zhao at UIUC, another Francis Arnold Lab alum, and Dr. Sriram Chandra Sekharam at University of Michigan. And the follow-up Zoom chat will occur via the link in the chat box. I can post it over there, and see you there soon. Okay, thank you so much.
Thank you very much. Yeah, I think I didn't notice that I took too much time for the talk.
No. The doctor, you also kind of take, a little bit longer than I expected. But your work is kind of, two great thing. Together, so kind of combining within that limited time is the most challenging, I guess.
Yeah.
So
Okay.
so if you click the link over there in the chat, so do you see the link? Another Zoom. So if you click and then if you kind of click another one, and then you will automatically move to another Zoom one, and then I will meet you there once I close this meeting. Did you find
okay.
the? And then you click that one, and then some people will joining us. To discuss more.
Oh yeah. Sure, I will do that.
And then we'll kind of discuss minimum 30 minutes more. To
Sure.
discuss about your exciting work and also many
Sure,
mathematics.
Yeah. Okay. Okay, yeah. So I will click that link. I think I will leave soon, right?
Yeah, you're automatically changing from this room to another room.
Yeah. Okay. Good.
Like
good.
kind of you
Okay.
flying in the sky.
Yeah. Okay.
Okay, so I will meet you there soon.
Yeah. See you soon. Yeah.
Okay, so Tony, thank you so much. Have a great weekend.
You're very welcome as always.
Yeah.
And you too.
Sure.
All right.
Take care.
Bye-bye.
Thank you.